All right, today we're going to be talking about phototransduction, how light influences action potentials, how we actually get the signals that light is hitting our eyes to our brains. And so let's jump right into it. For some basic anatomy, we have photoreceptors, rods and cones. Rods will basically differentiate between black and white light, and cones will help us see color. And so those are the difference. They're called cones and rods because rods are pretty straight up and down. They look like rods, and cones have a cone shape on the very end of their cell body. And those will attach to bipolar neurons, which will also attach to ganglion cells, which will finally send a synapse through the optic nerve. So that's important to know. So let's start up here. Within these cones and rods, we have these little discs. Both cones and rods have these little discs. And these little discs have membranes with proteins embedded in them and channels. And one of those protein complexes is rhodopsin. And what makes up rhodopsin is retinol and opsin. It's the combination of the two. Opsin is kind of a derivative of vitamin A. It normally has this cis conformation where you have a bend in its structure, and that's when it is active. And when I say active, it means that it's causing the opsin protein to be deactivated. Keep in mind, rhodopsin is a G protein coupled receptor. So down here you have your G protein called transducin, and it has three components or subunits. You have a gamma, a beta, and an alpha subunit. So what happens? Another thing you're going to have embedded in this membrane, you have phosphodiesterase, a ligand-gated channel, and that channel is going to allow sodium and calcium to diffuse across the membrane and depolarize the cell. And more on that later. So that's the basic structure. So right now we're set up. We have everything in place. When light hits this disc, or rather when it hits rhodopsin, it changes the conformation of retinol. And this retinol is going to change from a cis conformation, as mentioned before, and it's going to change into a trans conformation. So instead of bent, it will now be relatively straight. And what that does is causes a cascade chain to occur so that we're no longer allowing sodium ions into the cell to depolarize it. So we hit retinol with light, it changes the conformation to trans, and what that will do is deactivate retinol and activate opsin. And opsin is then it's G protein coupled, and so that G protein made up of these three subunits will now break apart, right? Breaking off the alpha subunit. That alpha subunit will then head over and activate an enzyme called phosphodiesterase. Now, phosphodiesterase is very important because it degrades cyclic GMP. Cyclic GMP is attached to this ligand-gated channel, which is allowing sodium to enter and depolarize the cell. In order to send off an inhibitory signal, we need to stop depolarizing the cell. In order to stop sending an inhibitory signal to a bipolar neuron, we need to inhibit this depolarization. So what phosphodiesterase does is it degrades cyclic GMP to GMP, and that's shown over here. So we degrade it, it becomes GMP, and because it's now degraded, it detaches from that ligand-gated channel and closes it. So now sodium is no longer entering the cell. Once that happens, we've completely stopped depolarization, and which causes that cell to stop sending potentials down its membrane and stop sending inhibitory signals. Those inhibitory signals are glutamate. The way that works is if we're depolarizing the cell, we're then allowing calcium to enter the cell, which allows us to bind to glutamate neurotransmitters and send them to the bipolar neuron. If we've stopped that process, then we're not sending inhibitory things. We can now send an action potential down the bipolar neuron. So before we can restart the cycle, we're still in a trans configuration for retinal. And that retinal needs to be bent back into its cis state for us to recycle. So to get there, there's a couple things that we do to reset. Phosphodiesterase will eventually deactivate and join back to its G protein, transducing all three subunits together. Retinal is going to go to a pigment epithelium and get rebent into its cis configuration, and then it's going to enter back into our opsin protein, forming rhodopsin again. And once all those factors have reset and phosphodiesterase has been inactivated, we're now back to a state where cyclic GMP is now active again because phosphodiesterase is not interfering, and we're allowing the cell to depolarize because sodium is getting in again. One more thing on retinal, we can't go through this cycle unless it's reset, unless it's gone back through the pigment epithelium and reconfigured to that bent state, and that takes some time. Even if, well, yeah. There was one more thing. While these do send off excitatory signals, I mainly focused on inhibitory signals, because if you inhibit a bipolar neuron, you're not sending any potentials down the synapse or down the membrane and then synapsing, such that you actually send a potential through to the brain. And so it's pretty important that we understand how light changes retinal such that there's a cascade effect, and we deactivate the whole thing. Deactivate inhibitory signals. So that is phototransduction, and that's how rods and cones interact with light. There are a couple nuances with rods and cones. Rods do black and white, cones do color. Slight differences, but in general they work the same. One just sends off signals for color, and others send off signals just for light. And that's it.